Okay, today we're just very excited to have Dr. Vernon Ansdell join us from Hawaii to discuss the very important and not very well, I think, publicized challenges and things that are happening right now with neuroangiostrongyliasis. So, and then we're going to have a little bit also by Dr. Will Sears at the end on the diagnostics and some of the specifically molecular diagnostics in that area. So if you want to take it away, Vernon, thank you so much for joining us.
Yes, thank you. Thank you very much for your introduction. Welcome from Hawaii. You just missed the sunrise, but luckily I didn't miss it. I was afraid I'd oversleep or something, but that didn't work out. Okay, so let's get moving on this. You'll see that I'm going to cover quite a lot of topics in this, but I'll be giving you some key references, if you like, as we go along. So very briefly, neuroangiostrongyliasis, or rat lungworm as it's used, as it's often called. It's caused by a nematode parasite, Angiostrongylus cantonensis. And actually, this is quite a recently identified parasite. It was first identified in rats in Guangzhou, in China in 1935. And Guangzhou at that time was known as Canton, and hence the name Angiostrongylus cantonensis. The first human case was in 1944 in Taiwan, Formosa, as it was known then. And as you can imagine, after the end of the Second World War, there was a lot of chaos, a lot of transport in the ocean, moving things around, and it basically set the stage for spread of the parasite. So it spread, we think, mainly in cargo ships by means of rats that were infected. And over the several years, it spread throughout most of Asia and into the Pacific. And the first cases in humans in Hawaii were actually in 1960. And over the years, cases have been reported from the Caribbean, southern part of the United States. More recently, there have been cases diagnosed in humans in South America, in Brazil, in Ecuador, and some other countries, and also in Africa, in West Africa, in Nigeria, Ivory Coast, excuse me, South Africa, and Egypt. And then really relatively recently, there have been cases in animals, infection identified in animals in Southern Europe, in the Canary Islands and Mallorca. So the disease is spreading. We think it's probably in areas now, and we just don't know. It's just not been identified yet. But there's every reason to think that it will continue to spread, including maybe some impact from climate change. And nobody really knows what's going to happen with climate change in this disease, but it seems likely to me that there will be new areas that will be good for the parasite. And some of the areas where it's established now may become less favorable. So we'll have to see what happens. A little bit about the life cycle. The natural life cycle is between rats and mollusks, slugs, and snails. The parasite lives in the heart and lungs of the rat and develops, and L1 larvae are passed in the rat feces and are ingested by the slugs and the snails. The parasite develops in the slugs and snails into L3 larvae, and rats become infected when they eat slugs and snails. So that's the basic sort of natural life cycle. There are reservoir hosts, or transport hosts rather, so-called paratenic hosts. And the parasite can infect these animals, but doesn't develop in them. So they ingest L3s, but it doesn't develop and cause problems for them necessarily. So monitor lizards, land crabs, shrimp, frogs. Centipedes have recently been identified as a parotenic host. They can all become infected, and it's yet another source of infection for humans. So humans become infected in three basic ways. The first would be by ingesting slugs and snails, raw or undercooked slugs and snails. That's one way that humans can get infected. In some parts of the world, that's easily the commonest way. Second way would be by ingesting raw or undercooked parotenic hosts. And the third way, and this is probably the commonest way in Hawaii, is by ingesting green leafy vegetables that are contaminated by slugs and snails. So in that regard there, you're very likely going to be eating whole slugs and snails, and by definition, they won't be cooked. One way that was quite a problem on the island of Maui was people were making smoothies out of, for example, kale. And it's very difficult on a kale leaf, at least some of the varieties. The leaf is very curly, and it would be almost impossible to sift through every sort of kale leaf and figure out if there was contamination with small slugs and snails. So people were making smoothies out of the kale and drinking that. That was very popular, considered to be very healthy. But there were certainly a few cases where it looked pretty obvious that the culprit was these kale smoothies. A little bit about the disease in Hawaii. First case was in 1960, as I mentioned, but it only became a notifiable disease in 2007. Before then, there were occasional cases reported. It could often go several years without anybody seeing a case. But from 2007, we have records, and on average, there were about 5 cases a year up until about 2014. And at that point, things started to go up. And in 2017, there were actually over 20 cases reported, and guaranteed we're missing a lot of cases, or for some reason they're not getting reported properly. So most of the cases are on the Big Island, the island of Hawaii, but recently there've been a few cases on Maui. And on this slide, that's in the sort of dark or gold coloring. We also have occasional cases in travelers, visitors, and these aren't on this particular chart. We think we missed a lot of those cases. So in 2019, there were some cases, but in 2020, obviously with COVID, there was a drop in the number of cases, only 5 in that year. This year so far, 2021, we've had 4 cases, and we're not halfway through the year, but we'll see how many cases come out. I think with COVID, it's not been a particularly important issue for the Department of Health to be trying to deal with. They've been, as you can imagine, swamped with COVID cases. A little bit about some of the reasons why we have Angiostrongylus here in Hawaii, and why the numbers seem to have gone up in the last few years. And one important part of this is a semi-slug called Parmarion martensi. They call it a semi-slug because it's sort of not a slug and it's not a snail. It's got like a half shell on the back. You can see it in this picture. I think this is the shell here. And this is an invasive species that originated in Southeast Asia. It first appeared on Oahu in 1996, on the Big Island 2004, and Maui 2017. And it seems pretty obvious to me that there was an increase in the number and severity of cases following introduction. We saw a few more on Oahu. We definitely saw a bump up in the numbers on the Big Island, Hawaii Island, and similarly on Maui. And there's a few reasons for this, but these are tiny when they're juveniles. They're very tiny, just the size of a grain of rice. So you could easily miss them on in a salad. and they're very likely, they're very susceptible to infection. So when they look at these slugs, more than 75% of them may actually be infected in the surveys. And when they do get infected, they build up incredibly high levels of infection. So they have often tens of thousands of larvae per slug. So if you eat one of these whole, uncooked, you could be ingesting tens of thousands of larvae, which is quite alarming. A little bit about the disease itself in Hawaii. It does seem to me that we often have a more severe illness than many other countries. For example, more severe than they see in Thailand and China. And it seems to introduce a disease that has more long-term disability and deaths than seen in many of the other countries. And in Hawaii, the severity is probably linked to the size of the infecting dose. So accidentally eating whole, raw slugs or snails in contaminated salads or ingesting them as a dare, which happens sometimes, you're going to get a sizable dose of infection. And remember that some of these slugs and snails, for example, the Parmarion that I talked about, and also giant African snails, can have very high larval loads. So you're ingesting an awful lot of larvae. Now, if you look in the textbooks, actually, you'll see things like, in the little section on Angiostrongylus, they say, well, most cases are benign and they're self-limited. They'll say things like complete recovery is usual. Simple symptomatic treatment is usually sufficient, and specific anthelminthic treatment is not indicated. And they kind of dismiss the disease as something, just a curiosity, but of no real medical importance. But you just tell that to these two women shown in the pictures here. On the left, we have she was a visitor, I believe from the West Coast somewhere. And she was on Maui, and she got really sick with this bizarre symptoms. Nobody could figure out what was going on. Clearly distressed, clearly ended up in hospital, and it was devastating for her. I don't know long-term sort of recovery stuff with her, but she was certainly ill for a long time and very disabled. And the woman on the right, previously young, healthy woman, and you can see her life now revolves around a wheelchair and a walker. She has long-term disability as a result of her infection. So this is a severe disease. Now, one of the things that really caught my eye about this was I went to what they have on the Big Island. They have support groups for people who are recovering from the disease. And I remember being very impressed by this is a totally life-changing experience for these people. They have long-term disability. There are a few people who die from it, but it's the people that survive, but they're left with this long-term disability. And for them and their caregivers, it's a totally life-changing experience. So that got me really interested. And I was thinking, what can we do to try and prevent this? People getting into this terrible situation. And I began to look at the treatment that people were getting and whether there was anything we could do to prevent these sort of severe situations for them. So let's just switch to an illustrative case and talk a little bit about a patient who was a healthy 42-year-old farmer on Hawaii Island, on the Big Island, and he presented with a 4-day history of headache. He'd never had a headache like this before. He had some nausea, but nothing else at that point. He hadn't traveled anywhere abroad. He was seen by his regular physician. Examination was unremarkable. He was diagnosed with a, quote, tension headache and treated with ibuprofen and muscle relaxants and told that he'd get better pretty soon. 3 days later, however, his headache was getting worse. He'd vomited. He now had electric shock pains in the arm and some sensory symptoms in the right leg. So pain in the left arm, other sensory symptoms in the right leg. So kind of an odd sort of mix of symptoms. He was seen in an urgent care clinic, and it was thought that maybe these weird symptoms were he was just anxious. So he was diagnosed as tension headache with anxiety and given alprazolam and told he would get better soon. So over the next 5 days, his headache persisted. He now had his pain in his left arm had gone, but he had numbness and tingling in the right arm. And this time he was seen at an emergency room. So he'd seen his regular doctor, he'd been to an urgent care clinic, and now he was in the emergency room. Exam was pretty unremarkable. He had a bit of neck stiffness, but they were concerned enough about him to maybe run a blood count, which showed a mild eosinophilia, which was sort of not really picked up on. They did a CAT scan of his head, I think trying to just reassure him that there was nothing wrong. But he was given narcotic oxycodone and referred as an outpatient to a neurologist. 2 days later, he was worse. His headache was really particularly severe. He had ice pick-like pains in his legs. He had night sweats. He had anorexia. He had insomnia. And by this time, he was getting really frustrated. Nobody seemed to know what was going on. They were dismissing him as being just anxious. And so he went to another emergency room. He was given some IV fluids, felt a bit better, and they told him they were going to discharge him and just keep his appointment with his neurologist. But he dug his heels in. And it's surprising how often people have to do this with this disease. They're not really taken seriously, and they have to become advocates, if you like, for their own health. So he refused to leave the emergency room. A good friend had told him that he might have lungworm and that he should get what he called a lumbar punch. And I guess the providers in the emergency room gave in. They performed a lumbar puncture, thinking, well, just do this just to keep him happy and reassure him. Anyway, they did the lumbar puncture. Fluid was abnormal, slightly turbid. Opening pressure was elevated. CSF protein was elevated. And he had 240 white cells with 27% eosinophils. So he had an eosinophilic meningitis. So at that point, they started taking him really seriously, admitted him to hospital, and started to take a detailed food history. And it was revealed that he often consumed unwashed leafy green vegetables from his farm. There were a lot of slugs and snails and rats on his farm. And they actually put all this together and said, okay, we think you do have rat lungworm disease. And they started him right away on high-dose corticosteroids and albendazole. And the diagnosis was subsequently confirmed by a positive PCR. So let's just make a few points about this, this illustrative case. The history can be very confusing. It can be non-specific. It can be bizarre. And symptoms are often initially attributed to anxiety or hysteria. And if you've got fragmented care, which is not unusual in sort of rural areas, that further delays the diagnosis and treatment. This patient went to, I believe, 4 different healthcare providers. There are often red flags, especially when you look back afterwards. The migratory symptoms are very odd, where symptoms seem to move around from limb to limb. They're asymmetrical. The onset of a new severe headache and the presence of eosinophilia, they should be red flags or clues that something is really going on. As you can imagine, it's not uncommon at all to get a delay between the initial symptoms and diagnosis. This patient, it was 14 days, but I've seen much longer than that, and some very, very frustrated patients. You need a high index of clinical suspicion to make the diagnosis early, and education about the disease is important, not only for medical personnel, but for the general public. In this particular case, it was actually, the diagnosis in a way was suspected by the patient's friend. And it was only because he was persistent and frustrated that he basically demanded to get a lumbar puncture. Sorry, I'm having trouble advancing.
Do you want to unshare and then reshare your screen maybe?
Yeah, okay.
Let's give that a try.
We're back in business, I think.
Okay, great.
So sometimes you get early prodromal symptoms as the parasite migrates from the gut to trying to get to the brain, but those are very non-specific. And just a few pointers about in adults, sort of thing that would be severe headaches, migratory limb pains, there may be cranial nerve palsies, there may be migratory limb weakness, and in more severe cases, bladder and bowel dysfunction. A little bit different in children where irritability and fever and abdominal pain seem to be more common. Some of this I've covered already, and in the sake of time, I will maybe move on from this slide, but important, and I'll keep emphasizing this, we think it's very important to start treatment with high-dose corticosteroids and anthelmintics as soon as a presumptive diagnosis is made. This means before the diagnosis is confirmed. Typically, we confirm the diagnosis with PCR, but that can take several days, and the clock is running, if you like. So we recommend treatment when a presumptive diagnosis is made. A little bit about cases in travelers. This is something I'm particularly interested in. There have been at least 80 cases reported in the literature, and I would think almost certainly there's way more than that occur that are simply not diagnosed or not reported. And I wrote a paper with a doctor from Thailand on this subject that was in Current Opinion in Infectious Diseases about 2 or 3 years ago. And we found these 80 cases, mainly in travelers going to Southeast Asia, the Pacific, and the Caribbean. And most of these travelers were from Europe, USA, and Australia. And the delay in diagnosis and treatment was common and not by any means unexpected. And there were a few cases in travelers to Hawaii. So this, I thought I'd mention this recent sort of outbreak, if you like, that occurred in Hawaii. It's really fascinating. We're in the process of writing it up as we speak. So these were 6 Tongan men living on Hawaii Island who developed Angiostrongylus after drinking Kava that was found to be contaminated by an infected slug. They found a macerated slug in the bottom of the Kava bowl. So one thing about this is that we know the precise date of exposure. We actually know almost the hour of exposure. And that's pretty unusual in this disease. They actually prepared the Kava in 2 large 5-gallon buckets, the sort of things you buy at a hardware store. The second bucket they left outside on the ground for 2 or 3 hours before the Kava was consumed. And they discovered this macerated slug in the Kava bowl, the wooden bowl that they drink from, after they really basically finished drinking that evening or that morning, it turned out. And with the epi investigation that they did after this, these cases, they found multiple Cuban slugs on the property and a single Parmarion semi-slug inside a garden hose. We don't, think a garden hose was the source of the infection necessarily, but it's possible. So incubation period in these 6 patients was short. It was 4 to 10 days. All of them had eosinophil when they were first seen, but we didn't exclude other causes of eosinophilia, so it's hard to know what to make of that. And the patients who had, there were 5 patients had LPs. One of them, it wasn't successful and they decided not to pursue that. They all had eosinophils in the CSF with their first LP, and but one of them only had 2% of 160 white cells. And of the patients who had LP, the PCR was positive in those who had an LP 14 days, and 25 days after infection, but it was negative when it was performed that little bit earlier. So 8 days, 11 days, 12 days after infection, it was negative. So I think that shows, it's not a large number, but it shows that, that at least in these patients, the numbers, that there was a switch at around 12, 13, 14 days where patients were negative, but then became positive. Briefly, something on diagnosis. Will is going to be talking in a little bit more detail about this. Several ways to try and make the diagnosis. Firstly, it's very rare to find larvae in the CSF, but it occasionally happens. Obviously, eosinophilic meningitis is the hallmark of the disease. Finding a positive PCR is confirmatory. There may be MRI changes in the brain and spinal cord, but these occur usually after about 3 weeks, so not particularly helpful. The findings can be suggestive, but they're not pathognomonic. And of course, peripheral blood eosinophilia may be a clue. Treatment with steroids is pretty much a given with these people. High-dose steroids, we use them in almost all cases that we see. Usually given for 2 weeks and then tapered if necessary. And again, treatment should be started as soon as a presumptive diagnosis is made. And steroids have been shown fairly clearly on more than one occasion to reduce the severity and duration of headaches. But there's no data on the prevention of long-term disability or death, which in some ways is the key thing, really. This is the paper from Thailand, Chotmonkhal in 2000 that really showed that steroids were beneficial, a nice study. And ever since then, I think people have used steroids. Treatment with anthelmintics is way more controversial. And as you heard at the beginning, some people say they're not indicated at all. But this impresses me. So you could argue that, if you can remove the small L3 larvae at a very early stage before they start to wander through the brain and the spinal cord, molting, growing, causing brain damage, that's a much better way to do it than to wait until they become much larger, L4 and L5 larvae. When they die, you'll get a lot of inflammatory response and presumably more brain and spinal cord damage. And if you look at numbers, L3 larvae are tiny. They're microscopic. They're 0.5 millimeter long and 0.03 millimeters wide. But the larger L5 immature adults, 12 millimeters long and 1/4 millimeter wide. So you've got a 1,500-fold increase in volume and a 2,000-fold increase in weight. That's a huge difference. So, in theory, get rid of them when they're very small before they've had a chance to grow like that. And the way to do that would be to use an anthelmintic. We don't really know about adverse effects of steroids. You could argue you're actually encouraging them to live and thrive, having a lot of steroids on board. So treatment with albendazole is controversial, been controversial for decades because of concerns that death of larvae would trigger a major inflammatory response and clinical deterioration. In fact, reality is that clinical experience shows that treatment with albendazole is safe, and it may actually be beneficial in the treatment of Angiostrongylus. And we do recommend, however, that anthelmintic drugs such as albendazole should be given with steroids until there's more conclusive evidence on safety. We have evidence in animals, but clearly not in humans. It'd be hard to do a study like that. One paper that showed that really the drug appears to be safe and certainly appears to be effective, at least in reducing headache, was this paper from Thailand. A lot of the papers about this disease have been written in Thailand. And unfortunately, they're talking about less severe disease than we see in Hawaii. And I believe less severe disease than we will see in countries as the disease spreads across the world. There's very good evidence in animal studies, mice that are similar hosts, if you like, to humans. There's evidence in mice that the drug works, but that it does unfortunately need to be given very early in the course of the disease. And the magic window at least in mice, is to give the drug within 5 to 15 days after infection. So that's very early in terms of being able to make the diagnosis and be confident enough that you're going to start treatment. And certainly we recommend that as soon as you think it's a reasonable possibility, you start treatment with not only steroids, but albendazole. This was a paper out of Taiwan, written back in 1988, but it's a very well-written paper, I think, and nice design study, and does show this very clearly, that if you're going to treat, you have to treat early while there's still L3 larvae, L4. Once they've molted and become L5s, they become much, much less susceptible to treatment. A few just general treatment comments. You may need, if you've got a patient with severe headache and they're persistent, you may need to repeat the LP. That seems to help. Start with simple analgesics. They may not work. You can use opioids, but try to avoid them for obvious reasons after the acute phase of the illness. They've tried other drugs such as IV lidocaine, Lyrica, Neurontin, and there's sort of mixed results with that, especially Lyrica and Neurontin, really not much help from those drugs. Cannabis, the cannabinoids, used a lot on the Big Island. It's hard to really know how effective they are, and we really need a good study to look at that. Things that do seem to benefit and are certainly pretty harmless would be acupuncture, psychotherapy, physical therapy, occupational therapy. These really do seem to have an impact, this sort of multi-pronged approach. And patients may need to see pain specialists. So, you know, that's pretty much a whirlwind trip through angiostrongyliosis. And if you want to see a little bit more in depth about what we've been thinking, what we've been working on, we had some guidelines published in parasitology earlier this year. And that goes into quite a lot of detail about this disease and what we recommend for guidelines for clinical management. We talked about doing evidence-based guidelines, you know, really, you know, big document on that. Two reasons we didn't do that. One was nobody really had the time and the energy to embark on something like that. These are very time-consuming publications when you publish the evidence-based guidelines. It'll take years to get them published, and we felt we needed to get something out there fairly early. And the other reason for doing it is, unfortunately, there's not really much in the way of evidence out there to help write a detailed document. So what evidence there is in this document, but it's not a high-powered evidence-based guideline document. So to wrap up my part of the talk, just a summary of the key points that I was trying to make. So this sort of non-specific, bizarre, confusing clinical presentation often results in delays in diagnosis and treatment. And it's particularly important in areas where infection is relatively new and unfamiliar. So things are improving when it comes to diagnosis in Hawaii, but there's actually still delays it's just so confusing for a busy clinician. And it's an important consideration in travelers, as I've mentioned already. It's an important consideration as the disease pops up in different parts of the world. Theoretically, at least, early diagnosis and treatment may be really important in preventing severe disease with long-term disability and death. And making an early diagnosis is really, really difficult. And we do recommend, though, that when you make the diagnosis, even if it's just a presumptive diagnosis, you go ahead and treat right away with both the drugs. So what we urgently need are new tests that will promote earlier, easier diagnosis. And as I've mentioned several times, the diagnosis needs to be made and treatment started within that magic window of 7 to 15 days from infection. And that, I think, will give the best chance of preventing long-term disability and death. So the good news is that we think someone has come up with a test that will make it easier perhaps to make the diagnosis, and certainly make it easier to make the diagnosis at an early stage. And this is, to me, very exciting. It's a breakthrough that we've been hoping for, looking for a while. And with that, I'm going to hand over to Will Sears, who will tell us about this, this new and exciting test. Thank you. And we'll have questions at the end, I believe.
This sounds great. Thank you so much, Vernon. Can you hear me okay?
Yes, I can.
Okay, perfect. Thank you so much. That was wonderful. I believe, can I share my, I believe you need to stop sharing your screen, Vernon, and I'll share mine.
Okay.
There we are. Okay, very good. Fabulous. So now let me see what, okay. So thank you so very, very much. Thanks to Elise, and Adrian, Hannah, and Vernon for giving up a bit of time here in his excellent talk to let me talk about the molecular diagnosis of Angiostrongylus cantonensis. First of all, just to get out there, there's also a antibody-based assay, but we're not going to go into that. It's a bit controversial, and I think for the majority of what we would see, at least here in the US, a lot of our diagnosis would be made molecularly. And so thankfully I got to be a bit of a part of this story, and very fortunate. And so I thought we would go into some of the things that us and our collaborators have done. So first of all, I just wanted to acknowledge right up front that these are the collaborators that have been extremely wonderful to work with, and without them, none of this would be possible. Now, I just wanted to start by, as just a quick example, just to set like a foundational idea of how to think, I've come to think about how you make a more sensitive qPCR assay. So let's say that you want to detect this sequence. Let's say, what might you target? Well, you could target, how about the word worm? You could, if you wanted to target the word worm, you'd have one chance to catch this sequence. But could we improve that if we perhaps found a more repetitive sequence? Maybe so. How about these letters ITS? Perhaps if we targeted that, it would increase the sensitivity of our assay. And those letters weren't chosen at random, because I just want to acknowledge Yvonne Kvarnström's kind of championing of molecular diagnostics for Angiostrongylus cantonensis for the past decade, and a number of other tests too. She's works at the CDC, and honored to have her as a co-first author on our paper. So the test that she developed and validated targeted the ITS1 region of the a cantonensis genome. When you send test to the CDC for PCR, this was the test that was ran with these primers and probes that's shown here. But still, although that was excellent work, but there was still a recognized need for a molecular assay with greater analytical sensitivity, espoused by a number of organizations with expertise in rat lungworm. And I just wanted to point out here that including the governor's task force in Hawaii, and of which Vernon is the chair of the clinical subcommittee. And so how could we do this? If we want, perhaps we could use bioinformatics to find an even more repetitive sequence. And that's what we did. And so using a bioinformatic technique, we found a sequence that was really highly repetitive and developed a PCR against that. And this is the manuscript. I'm going to get into the data just here quickly, but I just wanted you to see that it's available. It was published in CID a few months ago, and super fortunate to be part of this story. And so just briefly, the bioinformatic tool, you can do this at home with a little command line, a few commands on a command line, and a fast computer. But it's called RepeatExplorer, developed by plant researchers out of the Czech Republic. And Eric Dahlströmberger, the bioinformatician, ran this part of their project. And this is the readout. And I just want to point out here that Contig 3990 was predicted to be one of the most highly repetitive regions. And when you look at the sequence, the consensus sequence, this is what you get. I plugged this sequence into PrimerQuest with IDT and some other sequences and ordered, just ordered the primers and probes that are shown here in yellow, purple, and green, and then applied that to some samples. And so what we saw, thankfully, was that our assay 3990, after the cluster it was named after, the Contig it was named after, was about, it became positive about 10 CT values sooner than the ITS1, pretty much across all different genomic DNA amounts, and including if you look at just the number of L3 larvae. And just so you know, the note that here the CT value on the x-axis is inverted, and so we did it that way so that just to appeal to the intuitive sense that bigger is better. And so the higher the bar, the more sensitive the assay. ACAN is in red and ITS1 is always in blue. And so we then looked at specificity, tested against clinical samples from Balis ascaris, Naphistoma, Teniasolium, Toxicara, and then also some other species of Angiostrongylus, the genus. And there was cross-reactivity with this nematode Angiostrongylus maceraceae, but this is confined to a very narrow strip in Australia, and it's not thought to infect human beings. The ITS1 assay also picks it up, although Jan Slapeta swears that this is actually a different species, and he is the expert, so I believe him. But I don't think it's going to hinder the use of this test. And then this is kind of my favorite figure. It's just because, so Yvonne Kvarnström is responsible for this figure mostly. But when she looked at her repository over the years of Angiostrongylus cases, she was able to pick up 15 new cases over the ITS1 assay. And then also this in part one of these was the first case ever confirmed from the state of Florida. So I think that's really cool. And that was the basis of the development of this new diagnostic that Vernon so kindly mentioned. And we've gotten back some positive, there have been a few labs implementing it with some good success. And so we're very excited by that. And for the last couple minutes, I just wanted to mention a little bit of other work I've been doing with molecular diagnostics, modern ones. real, I think, kind of the holy grail of some of these molecular polymerase DNA detection methods is having something isothermal that you could do in the field. And there are a number of them out there. Some people might be familiar with LAMP. But this is the one I've been working with, Recombinase Polymerase Assay, and it's very similar. And really quickly through this, it just uses a forward primer and a reverse primer, but instead of using temperature to de-anneal the strands like a PCR would with its thermal cycling, it just uses a recombinase to just kind of get in there and push them apart. It's from a bacteriophage. And so you don't have to vary the temperature, and it runs well at around 40. And so this is some of our results from the RPA that we have been running. And you, oops, sorry, there we go. And you can see it has a similar limit of detection of compared to the PCR. And then took this idea here from a COVID RPA paper where they visualized using a blue light reader or a blue gel reader with an orange filter. And you can actually visualize this not as robustly as, say, I would like without, the camera, but still it's a starting place. And then I also ran it on some clinical samples. And although some were, like, again, not as robustly positive as we would hope, there was still fluorescence up above the negative controls where you could call this positive with fluorimeter, and it didn't cross-react with Toxocara and athostoma. And then finally, just as proof of principle, we looked at different temperatures that this could run at and compared the different amounts of DNA and how quickly this test became positive. So if you look here at 40 degrees, it becomes positive. The double plus, means robustly positive, bright enough to really tell. At about 20 minutes or so, and 35 degrees, it takes it a bit longer, but it gets there. And so then I was thinking, well, how about body temperature? And so I just this was what I put in my shirt pocket. Now that's a shirt pocket underneath an Old Navy sweater, if to be specific about the methods. But it was just in my shirt pocket for 30 minutes, and then I looked at it under with the gel reader and took a picture through an orange filter, and you can see that it is you have down to 1 femtogram per microliter, which was limited detection using the fluorimeter and all these other things. So I don't know that I think that these more modern molecular assays, they're the future. I don't know if this one will necessarily be the assay, but I just wanted to introduce this here because I think there's going to be more and more talk of this as time goes on about PCR alternatives introduced to the field. But that's really all I have, except to thank all my collaborators again.
Thanks, Will. Thank you, Vernon. That was great for both of you. Let's see. I think we have one question from Scott Norton. Well, a few questions from Scott Norton. And so he wants to know, what is it about slug slime that causes infection in humans? Are larvae excreted in what the organ that produces the slime, or are they contained within the snail feces that contaminate the slime? I think this is for Vernon.
Yeah, so I'll try and answer that question. That's a great question, Scott. And the simple answer is there's a lot about this we don't know. They've found larvae in slime, and it's often thrown up as, this is how people are getting infected. They're inspecting their lettuce leaves, and maybe they're even trying to wash them, but they still appear to be getting infected. So it must be in the slime. And when they've looked, they find a few larvae in slime, but not really enough to make people ill, and certainly not enough to create the sort of illnesses that we see in Hawaii. So somebody is looking at this sort of as we speak to really try and figure out what is the truth about this. Is it a problem or is it not? To try and resolve that. In terms of where the slime it sort of originates, when the larvae get in there. I can't answer that part of the question, I'm afraid, but unfortunately, we don't know enough about this to really give you a precise answer. But there is some work going on right now, and hopefully maybe by the end of the year, the question can be resolved. Somebody in our group took a look at this a few years ago and published on it, and they reviewed a lot of the literature and decided that there really wasn't evidence that slime was a contributing factor, but we need to clarify that completely. Does that answer everything in the question?
Sure. I have one question to piggyback off that one, namely that can we assume that if you cook the slug, it will kill the parasite? And specifically, I know there's at least one city in the United States that has a slug fest where they have people, including famous local politicians, that come and consume slugs.
I think the evidence is that if they are cooked properly, thoroughly, then it will kill the parasite. And the same would apply to freezing the parasite if it's done properly. And it's a little bit more than just sticking it in the freezer for 5 minutes. But that should solve the problem. The problem is that often in restaurants, they'll cook this up, and if they're busy, they're not really cooking it thoroughly. And I think if it's sort of a little bit sort of on the undercooked side, they would tend to be a little bit more palatable, if you like. Once you overcook slugs and snails, and also some of the seafood produce, it becomes very rubbery and not very pleasant to eat. So there's a tendency to undercook things, and if they are undercooked, then you can get infected, because some of the parasites will be killed, but not all of them. And this seems to be a part of the problem in Thailand. And one of the things they do in Thailand is they have raw snails, and instead of cooking them, they stick them in a sort of marinade of spices and oils and things like that. And the idea is that this will kill the parasite. But there's a very good paper out of Thailand that looked at this, and they showed quite clearly that it'll kill some of them, but not all of them. And it's all these sort of odd things like this that may explain part of the reason why cases in places like Thailand tend to be milder. There's other reasons as well, but that may be one of the explanations.
Okay. Yeah, great. Actually, and then Amy mentions in West Africa, they routinely eat sea slugs, but cook them for a very long time for this very reason. Is that true? Can Angiostrongylus survive in sea slugs as well? I guess I thought it was more of a freshwater thing.
Yes, that's my understanding.
But
so this is Amy. The sea slugs there live in what's called brackish water. So
brackish water.
They're kind of a hybrid. But I can tell you, having unfortunately had to eat them, that the person that made them for me told me they had been cooked for 24 hours and I shouldn't worry about Angiostrongylus.
Nice. Okay. That is nice.
Yeah.
Okay. And then one other, well, there's a few questions. So is there any evidence so far that climate change is affecting the distribution of the parasite or the vector?
I think we're starting to see evidence of that. Yeah. There's somebody in Hawaii that's looked at that, and some other people who've done stuff over the years. And definitely it looks like there are newer areas that are opening up, becoming a better environment for survival of slugs and snails, and rats seem to be almost everywhere. But then at the same time, I think you have to admit that there may be less disease in some areas where the climate becomes less favorable. We just simply don't know really at this point.
Okay. And then Scott Norton made a comment about the fact that the kava is made differently in different places, and the kind that's dried kava roots, it's interesting that it was really the preparation and the water source, as you mentioned, was the contaminator factor there. Although in other areas there's roots that are directly made right out of the soil into fresh kava and have been associated with amoebiasis. His question is, in humans, does infection always involve the CNS, or can it involve non-CNS organs as well?
That's interesting. We think about a third of the larvae end up in the brain and the spinal cord, and the rest of them just die on the way there. Some of them end up in the skin, and you occasionally get patients will have a history of a rash and itching. Some of the parasites can end up in the kidney, and if you check the urine, you may see microscopic hematuria, but a lot of them just get trapped in the various other organs as they try and make their way to the brain. So as far as we know, they die. They really need to be in brain and spinal cord to be able to find the sort of food they want to be able to live and thrive and molt and grow and so on.
Great.
I have one other question for Will. Will, do you have data on the sensitivity of your novel PCR depending on the timing of when the CSF was collected, or not yet?
So I'll honestly have to look back at the paper. We pulled out the timing because they have documentation through the CDC about the timing of when these samples were sent in. But I forget the earliest time point that it was done, but I hope it's different labs. But I remember it wasn't within 7 days or so, but I would be interested to see, especially with Vernon's assay or the group that drank Kava and stuff, if it was applied to that, where would it be? I think we'll find out more. It's time.
This is Tom. Can I interject?
Yeah, please do.
qPCR was able to pick up in a couple of the patients from the outbreak in Jamaica, the 3 or 4 days earlier than was picked up at the CDC at the time using the qPCR. So it was negative. This is one of the patients that was negative. Let's say, I can't remember, day 8 perhaps, and then positive at day 12. But this new PCR picked it up on that day 8 or 9 or whatever it was. But nobody's really looked, I think, that earlier than that. From what I remember.
And is the CDC adapting now their assay to this new set of primers and probe? Is that happening, or?
I haven't asked Yvonne exactly what they're using now. I mean, I imagine they would run both, but I don't know.
Okay, it's past 1. Should we?
All right. Well, thank you so much to Vernon and Will Sears and for the questions, and we really appreciate it. This will be on our YouTube site for everybody to view again if they want. This was very, very helpful, very informative. We really appreciate you bringing some attention to this topic. Thank you.
Thank you.